All right. Well, we're starting to get nearly a full screen, so I will start to kick us off by welcoming everyone. So, for anyone that is attending this call for the first time, we hold this industry-wide call every month for any company really that wants to participate in the cultivated meat value chain. And the purpose of these calls is to introduce and welcome new solutions providers into the industry with the end goal of establishing new collaborations that can help to accelerate R&D and commercialization. And so if your group is interested in presenting on a future call, or maybe you're working with a company that could benefit from being on these calls, please do feel free to put them in touch with me directly, and we'll be happy to work them into the schedule. And then we'll turn it over to Zachary Sun from Tierra Biosciences, who will be discussing their cell-free protein production platform. So as always, you can feel free to drop questions in the chat throughout, and we can get to them at the end of each respective talk. So Zach, over to you.
Great, thank you. Great. So my name is Zach. Thanks for inviting us here. We are at Tierra Biosciences producing purified custom proteins. So really a tool provider for not just synthetic meat, but also kind of other like ag fields, pharma fields, industrial biotech fields. So, wanted to kind of introduce ourselves to the industry and also, right, learn as well about the things that we can help you guys out with as you guys grow. So, with that said, I'm the CSO and founder here. Here we think, proteins designed by humans, nature, or AI. We want to be producing them to help scientists kind of translate their protein designs into material reality by enabling exploration, engineering, and production at scale. So that's really our mission here. When we think about protein production, right, many of the recombinant protein production, many of the standard ways today are cell-based. So when you think about cell-based protein production, really you're trying to solve a biology problem for every protein that's made. And so with every protein that you'd like, right, typically you're setting up a team, trying to make that protein. And if it's made, that's great. But when it's not made, right, there's not much of a learning cycle. And so you kind of try until you get the protein. And so this can take 4 weeks up to months, and can be quite kind of laborious and time intensive. And so the Tiara solution is really to explore a different modality of protein, which is cell-free protein production. In this case, proteins, digital sequences just can be sent to us. And then we have a factory I'll go over where effectively we use cell-free systems. These are lysates that if you put in synthetic DNA, they will make protein, but there's no cell involvement. And so when you do it this way, you can start to massively parallelize kind of the activity making protein and make proteins across different kind of fields in the same way. So it gets you more biological space. It's not a living cell. And so things that are toxic or things that would be hard to make recombinantly, you can get access to. Again, you can scale this up very quickly, both in the number of proteins that you make and also in the amount of proteins you receive. And then you can also start to learn a lot from the proteins made. So our value propositions with our customers are effectively, we can help them convert, 100-fold complex designs 10 times faster into high quality protein. And so people really use us to, get a lot of throughput. So test a lot of protein very quickly, find their candidate, and then scale that candidate up. We'll go over now kind of some of the kind of platform, then the technology, and then kind of some of the applicability to the ag or to the cultivated meat space as well. So to order proteins, we've built a online portal. Here you can pretty much log in and then start ordering at the microgram or the milligram scale. Proteins starting at $5.95 with volume-based discounting. So we really tried to make this as easy as possible to really abstract away a lot of that, how do you make a protein? How do you have the equipment and such? And so just with a digital sequence, right, you can get started. And then we give some feedback related to kind of manufacturability risk. One thing about cellular manufacturing, right, is it's quite unpredictable as to if you try to make a protein, like, will that work or not? In cell-free, there's still a level of unpredictability, but there are definitely features that we've identified, right, that make manufacturability easier or harder. And so that feasibility helps you kind of with additional information before the order is placed. And so there's a nice video online if you'd like to see how that portal works, but you're always also welcome to just log into the portal and try it out. So what we deliver is effectively proteins and data in 3 weeks on the microgram scale, 4 weeks on the milligram scale, and the deliverable is physical and digital, right? So for every protein, we can try it up to 4 conditions in our standard kind of workflow, and we'll give you a data sheet with, the protein's concentration and what was delivered. And so these 4 conditions are effectively taking the protein, tagging it on the N terminal and the C terminal, and then trying different cell-free systems that have different properties. What we find is that when you do that empirically, you start to identify the right kind of parameters to make a protein, right? So that's really important for debugging and increasing yield. And so each one of these proteins is also physically delivered, and so that's on the bottom left. And then finally, I think this kind of goes to the trying different conditions, and this is unique to cell-free kind of manufacturing. With every protein, we'll deliver a page shell kind of showing the manufacturing of each variant. And so in this case here, this optimization analysis that's kind of built in. For the first protein, it looks like it behaves best on the N termini. The second and third protein probably behaves best on the C termini. The fourth protein is about the same either way. And the fifth protein, you really want to tag on the N termini, because if you tag it on the C, you'll get lower yields, right? And so this kind of optimization analysis really helps when you're debugging especially hard to make proteins, try to figure out what's needed, or also increasing yields of proteins, right? The end protein delivered is virtually the same. It's just off by a couple scars. We don't include the infinity tag that we purify on when we return to you, but effectively that makes a really big deal, just that one change. And then the product, right? We have a standard microgram and milligram scale. You can find these specifications on our website. But the microgram scale gets you up to 50 micrograms, depending on the protein stability. The milligram scale can be up to 2 milligrams. And again, we return everything as purified protein. We also have a gram scale, so if needed, we can scale up the amount of material made up to 10 grams. And we can make things really small to like 3,000 amino acids in size, right? And so really the whole spectrum of recombinant protein production we can cover. Our systems today are mostly based on E. coli. So basically, E. coli systems have been adopted, and we'll show you how they are different in nature to make different types of proteins. A lot of our R&D is going into expanding the repertoire so we can offer different types of biochemistries moving forward. So a bit about the technology here. Really, when we think about cell-free manufacturing, it's really a comparison of biology versus chemistry, right? And so cellular manufacturing, right, we think about as biology, right? There's stringent DNA requirements, cells talk to each other. They don't really want to make the recombinant thing that you want to make, and so you have to battle that evolution component. And there's this thing called a cell wall, right, which makes it really hard to get things in and out. And so this matters a lot if, for example, you're trying to make a hemoglobin, right, and you need to supply excess heme. To get that through a cell wall can be quite difficult, right? So how do you provide that to increase your yields? And so, when we think about kind of scaling cells, I think all this complexity comes into the fold. And so what we said at Tiara was, okay, biology can be pretty hard to engineer. It can be pretty hard to scale. So can we change it into chemistry instead? Can you go from effectively digital sequences to physical protein and never have to touch a cellular layer? Because if you can do that, then you can take advantage of automation, liquid handling, predictability, and start to kind of abstract away some of these problems, right? And so for us, cell-free manufacturing is just that, right? It allows us to use input DNA that's easily programmable. It allows us to make multiple variants of proteins very quickly. Easier engineering, so there's no cell wall, right? So we can add substrates as we need. You don't have to fight evolution anymore because there's nothing living. And ultimately, right, highly scalable automation because it's just a liquid cocktail that we use. Another way to look at this is just kind of when you think about the scale up, right? In the automated biology world of cell manufacturing, you've got DNA picking, growing cell mass, lysine. You're solving more biology when you scale up. And so realistically, right, you have around a 10-foot higher like capex and square footprint than you would for a cell-free process where you're literally just scaling robotics and liquids, right? And so I think the great example of this is on the right here. On the right, you see is GFP that we're making in gram per liter titer. To make that amount of GFP in a cell-free process, we are taking out liquids from a freezer, combining it together, adding DNA, and you get GFP. Whereas if you want to make the same amount, in a cellular process, you've got to stand up a large fermenter, right? Have that CapEx ready, and then build from there. And so it can be a really efficient manufacturing process as well as a fast manufacturing process because it's very quick to go from small volumes to large volumes. I'll show some data there. The actual platform that we have at Tiara really wraps cell-free manufacturing around automation and AI to both, again, deliver physical protein and digital insight. And so what we do at Tiara is take digital protein sequences, we'll convert that to physical DNA, and then we'll attach pretty much different N-terminal and C-terminal tags to effectively make different variants of your protein. That allows us to then go to a cell-free synthesis stage where we can do multiple small reactions effectively to identify what is the right condition to make your protein, purify those, and then analyze that, get that back to you as physical protein in the mail. The AI piece of this comes from making many proteins, right? So while we make proteins, we also learn from that, and that improves kind of the predictability of the next protein made. And I think this here is an important slide because it really shows how you can go quickly from a research question to identifying what you want to very quickly scaling it and then moving forward with pilot production. And so this is obviously just GFP. What you're looking at is lysate, where we've just started not bright green, but we just put in our DNA, waited up to 16 hours, and then you get this bright green color. So we can go from the smallest tube, which has 10 microliters, to a milligram, microgram and milligram scale up to gram scale. And these jumps, right, can take only a week because once you identify the conditions to make a protein, then you can very quickly jump to the next scale by just adding more of everything as opposed to having to solve biology again, right? And so this is quite useful for our customers because they're able to go from these kind of, I think, I want X screen to quickly testing X and then scaling up X for further testing. And so it's really scale to fit, right, to help really push the R&D process forward. So in the final part of the talk, I'd like to go over kind of the protein classes that we can make and some classes of applicability to cultivated meat as well. So we've made many types of proteins from, again, really small things to really big things. This is just a sample smattering of those type of proteins that we've made. And so on the left here, we've got these pretty complicated cyclic peptides. So these are multi-domain kind of pathways where you get a pre-pro peptide, it gets knotted and cleaved. So we're able to make these in our system, and then these have the correct activity and topology, which we can verify by kind of activity assays in this case, and also by mass spec data. Interestingly, you can't chemically synthesize these peptides, so they're really only biologically accessible. So that's on the very small end. When you think about kind of the middle area, right, Cas enzymes are a good example. We can produce complex Cas enzymes, toxic things, and other enzymes as well. And we can show that those are active in our system. And so in this example here, we've got a Cas RX. We show it degrades RNA appropriately, and also right is made in pretty good concentration. On the right, we can make different kind of targets, right? Again, varying from really small to really large. Here, the gel we're showing is effectively a campaign we ran for a customer, looking at different types of targets. And you can see this is a in lysate gel. So you can kind of see that, kind of think about a Western. So you can kind of see those targets being made in every lane. One of the unique, I think, pieces of the cell-free systems we have here is that it's not just limited to just proteins from E. coli. And so, right, when we think about E. coli, we typically think, okay, well, enzymes, but probably not growth factors, probably not cytokines, definitely not, full-scale antibodies. But what we can do, because it's an open system, is we can play with the chemistry to effectively make it more oxidizing. And so we have two kind of systems we have. Took us around 5 months to build kind of the second one, so you can see how fast cell-free engineering can be. On the left-hand system was our traditional enzyme system, but we fed it a bunch of biologic kind of modalities. We can make 6 of 14 of these. But on the right-hand system, after we make the system very oxidizing, now we can make all 14, right? And so what you're looking at here is effectively soluble protein in lysate. And so again, it's kind of like a Western type technology here that you see. So as this applies to kind of cultivated meat, right? The kind of one of the areas that we're excited about is in the growth factor area. And so, right, as we're expanding the the types of kind of medias that are being produced and the types of cells that we want to culture, is there kind of a interest space in quickly prototyping, testing, and then scaling up different protein variants, right, for testing. And so what we're showing here is a campaign that we ran internally where we wanted to try to make some of the most commonly used growth factors and produce those again in parallel in 3 weeks. And importantly, identify what is the right condition to produce these to maximize yield and expressibility. And so on the table here on the left, right, are some of the ones that we've made. If they're not marked, then they're human. Otherwise, one from chicken, one from mouse. What we did was we effectively shuffled our tag, kind of expanded it past the traditional ones that we use, and also kind of the conditions to say, well, what's the right condition to make this protein? And then what are the titers that we're getting? And so on the right, you can see kind of for albumin, right? This is a 17 disulfide-rich protein. So from the, like if you look at the biophysics, the structural piece of it, it's pretty complex. But we're able to make this in our biologic system and make it at decent titers. So around 0.24 grams per liter. And on the bottom is a good example of like why when you have an open system, you can start to increase titers in a way you can't do in cells very easily. Here you have hemoglobin and myoglobin, right? They require heme groups and subunits to come together. So for the hemoglobin, we can both express both subunits together. And then this is a reducing gel, so you can kind of see both subunits there. And when you add heme in, right, you can increase the yields of the production. The same occurs with myoglobin. You can increase the yields by adding heme in exogenously. The second example here would be like for a traditional growth factor, if you want to vary the species or even do protein engineering against it, again, that's a 3-week process, right? And so here, what we've done is we chose 29 FGF2 variants and quickly produced these in this kind of a parallel process. We were able to get concentrations of up to, gram per liter in a purified form. And these have been customer validated in cell culture, right? So they work, are active and non-toxic. And so again, a great example of how, if you're looking to expand past your traditional kind of proteins, that you can quickly test new ones, learn some insights about kind of what's working, what's not, and then quickly test those and then scale up, right? So some of these, for example, duck, goose, pheasant, quail share the same FGF2 variant, right? But other ones are quite different. And so you can see from this table here, at least, some produce quite well. Others, right, like elephant, turkey, right, are lower yield, right? And so there's some interesting biology there to glean, but nonetheless, right, can be you can quickly test and scale these. This is a little bit kind of more in the biologics world, right? But you can also start making antibody modalities. So if you need kind of to stand up an antibody or an scFv to do some testing, right? These can also be made in the system. What we're showing here is a monoclonal antibody. This is really a proof of concept, right? Just in case that you can really get complex here. 16 disulfide bridges, 4 subunits, heavy and light chain. And so we're able to make this classic trastuzumab monoclonal antibody in the system, and then we can test it against ELISA for activity. The yields, we can make pretty good yields of the kind of antibody modalities, so CFEs, Fabs, VHHs, antibodies, that type of stuff. So those are definitely accessible within the system as well. And the final slide I have here is just kind of some case studies of how we've enabled some rapid innovation for our customers. And so one is some work we did with Salesforce. A lot of the LLM work today, right, on protein design to make a more stable protein or a more active protein, right? Really interesting area. But one limitation of that area is that you can do many designs of proteins, but you have to be able to produce them to test them, right? And so we built a collaborative work with Salesforce to effectively do this on lysozymes, LLM trained model. And the goal was to keep the activity of the enzyme, but take it far away from anything that you would see in nature, right? And so proteins were designed, we then synthesized them, right? For the paper, we show 200 synthesized proteins. These proteins were then tested at Tierra, and the winners were then crystallized and tested, made in cells, and then crystallized by our partner at UCSF. We ended up finding a candidate with 31% sequence similarity to wild type enzyme that had activity and was crystallized. And we co-authored this work in Nature Biotechnology, so released it in January, so you can see that paper online. On the right is kind of in the ag space, right? For a Global 500, they wanted to develop kind of a protein-based insecticide. Tiara was the only one that could really produce these all the candidates. And then you can quickly do kind of dose responses to get phenotypic validation, just showing both the enzymes are active, and it kind of establishes a quick screen pathway. So that said, would love to learn more about the, what we can help the field with in the research and scale up needs. So feel free to email me, and happy to take questions.
Thanks, Zach. That was a great presentation, great walkthrough of the technology. And yeah, I guess the speed is definitely on display here, because I think we chatted over the summer at some point, and now you already have a bunch of data from a bajillion species and different proteins. So that's great. All right. Well, I'm not seeing anything else. Thanks to both the presenters today. Really interesting talks that I think can add a lot of value, especially at this early stage of industry development. So hopefully some folks get in touch with you over email, and maybe some new collaborations will be formed. So thanks, everyone. We will see you in the new year.